Hi, my name is Daniel Blackburn, and this is our second video from Plant Soil Environment course. In this video, you will learn about what is a soil nutrient, what are the criteria that we define if this nutrient is essential or to plants, and what are the factors affecting plant uptake of these nutrients and soil fertility in general. So this is an introductory course that we start up before we deepen these subjects even further. Alright, so this is the second video from SWAE4401 Water Nutrient Cycling in Soil Plant Environment. We will talk about nutrients today and factors from the soil perspective that affect soil fertility in general. Okay. So first of all, how do we came about knowing how plants take their own nutrition, how plants eat, how plants take the minerals from the soil, and what are the elements that makes plants. So this dates back from the origins of the basic analytical chemistry, when we start analyzing things for different elements, when we figure out there were elements and we figure out how to analyze for them in different media. So when we start analyzing soils and plants, we figured out that some of the plant composition was very similar to what we would find in soils. So there is this idea that plants were taking their nutrition from soils, especially when you analyze plant ashes and you analyze the ashes from soil organic matter also. So soil minerals will be present also in the plants, and that led for this initial theory that plants were drawing their nutrition from the soil. And in this author in the 1800s, he was hypothesizing that the more minerals you have on the soil, the more fertile the soils are, the better for the plants they are. So they were already talking about the mineral nutrition of plants, but they were it took some time to figure out with Justus von Liebig in 1840 the law of minimum. The law of minimum is regarding that it's not about how much nutrient you have on the soil, how much minerals you have on the soil, but is actually what we should care about mainly is which mineral is more deficient. So what is the most limiting factor on the soil, and that goes not only for nutrients but any other thing that affects plant growth. So for example, if it's water that is missing on the system, that limiting factor, water, will affect plant growth and will limit the maximum growth of the plant. So the most limiting factor will be the one that is limiting the maximum performance of the system overall. So this is an explanation here on the barrel, showing that the lower you have the limiting factor, that will be the conditioning of what is the maximum performance of that system. So if it's a nutrient like nitrogen or phosphorus that is deficient and it's the most limiting factor on that system, that nutrient deficiency will condition the plant growth. Doesn't matter if you have a lot of overall, the amount of minerals on the soil is good, the limiting factor will be the one that it's conditioning plant growth. So this is the law of minimum, very important. Then we start understanding what is the chemical nature of these minerals that the plants will take up. What is the chemical nature that is available for the plants? And curiously, although we tend to think that hydroponics is something very modern and very recent, it is actually as a form of cropping, but hydroponics was one of the experimental ways that we figure out that plants actually were having access to actually inorganic ions on the soil. So inorganic ions are through hydroponics, we figured out in the 1800s with Julius von Sachs and William Knopf that actually the nutrition of plants was through the root absorption, or could be leaf absorption also, but the absorption of ions, mineral ions, not organic molecules. So soil organic matter is not a nutrient for plants per se. Actually, when it's mineralized into inorganic ions is when the plants can take up these nutrients and have access to them. So by using hydroponics, you can modify the solutions, add different forms of these chemicals and of these minerals, and see if the plants can use these minerals on that form or not. So in this way, we could study and understand what are the available forms, chemically available forms of these minerals to plants. Now, when we start doing this, we have these two fellows here, Arnold and Stout, in 1939. They coined the criteria, whereas we define if a nutrient is essential to plants or not. So Arnold and Stout in 1939 defined this criteria. The first criteria is the nutrient must be required for the completion of the life cycle of the plant. If you do not have this nutrient on the system, the plant will not go from seed to seed, will not complete the life cycle. Second criteria is this element must not be replaceable by another element, which is the case, for example, of silica. So we have silicium. It's a nutrient, but it's not an essential nutrient, because if you do not have it, other nutrients like calcium can take the physiological roles that they have on the cells. Even though you may have a lot of silica on the plant materials, this is not considered an essential nutrient because it may be substituted by another element, so it's not essential. And the third and final criteria is that this element, this nutrient, must be directly involved in the plant metabolism, or it must be required for the completion of a specific physiological function. So if it fulfills the three criteria, it's considered an essential nutrient, and if it does not fulfill, it's not considered an essential nutrient. It does not mean that some nutrients can be even useful or important for plants, but in some situations not considered to be an essential nutrient. Okay. So here we have the composition of animals and plants. This is an overall general case, but of course this varies a lot within different plants and will vary quite significantly also among different animals. But you may notice there is a big difference between the composition of oxygen, carbon, and hydrogen, and plants will have quite a lot more carbon than animals. It means this is because plants will have a lot more fibrous materials, carbon-based materials, whereas animals will be based more in proteins. So because of that, it will have less carbon in their composition than the plants. Nitrogen, we will have also much more nitrogen in animals than we will have in plants. That's the reason also because animals will contain a higher content of proteins. Calcium is higher in animals because of bones, and that's similarly for phosphorus. Phosphorus is shown in much lower concentration in plant materials than it is in animal materials because the bones have calcium phosphate. So this is why phosphorus is so much more abundant in animals than in plants. Potassium is a little bit higher in plants, but this can vary quite significantly in plants also. Sodium is not an essential nutrient for most plants, so you will not find it in high amounts. Then you can follow up the list and you will find all the 16 and other 16 essential nutrients, but other elements that are not essential also may be found in this list. Yeah, one of these non-essential nutrients, the most important one, most significant, is silicon here, that you will find in high amount in plants, basically being part of cell walls, but it's not considered essential, as I told before, because it can be substituted by calcium in this cell wall function. So here they are. These are the essential nutrients. Yeah, we have the basic nutrients, the primary macronutrients, the secondary macronutrients, and the micronutrients. In total, they are 16. The basic nutrients: carbon, hydrogen, and oxygen. They are marked here by their classification on the periodic table. Carbon, hydrogen, and oxygen, they are the basis of all organic matter, all life on Earth. And we do not say they are nutrients per se, soil nutrients, because they are being acquired from water and air for the plants through the photosynthesis. They are not being acquired necessarily from soil solid phase. So they can be acquired from the soil air and from the soil water, but not from the soil solid phase. So these primary macronutrients and secondary macronutrients and micronutrients, these are the ones that are coming from the soil minerals. Yeah, these are coming from the soil solid phase in general. What the soil is providing for the plants. Yeah. The other nutrients, carbon, hydrogen, and oxygen, they're coming from water and air. So the primary macronutrients, nitrogen, phosphorus, and potassium. The secondary macronutrients, calcium, magnesium, and sulfur. And the micronutrients, iron, manganese, zinc, copper, boron, molybdenum, and chlorine. So why these are separated in these categories? The macronutrients are consumed in larger quantities, and therefore they're needed in higher concentrations in soil solution. So because you need them in soil solution in higher amounts, this is why we separate them in order of importance. But you may see that sometimes their order of importance as the concentration we need them in soil solution is not exactly reflected in the composition of plants. That is to say, for example, that phosphorus is found in much lower concentration in soils than calcium, for example. But we say the phosphorus is a primary macronutrient, whilst calcium is a secondary macronutrient because we need a lot more phosphorus in the soil than we need calcium. So calcium will be classified together with sulfur as a secondary macronutrient, while phosphorus is classified as a primary macronutrient. All right, continuing. So these elements, the plants need them. They're essential for plants. Without them, they cannot complete the life cycle. They cannot perform some essential physiologic function, and these elements cannot be replaced by other elements. But there is some range of concentrations that it's optimum. when you have this range of concentrations, the plants respond better. Yeah, and in some cases, if you have too much of this nutrient, what will happen is they will be toxic. Yeah, will be toxic to the plants. So we have this, that the nutrient can be in concentration in the soil where it's so low that the plants starve for that nutrient. And if they starve for the nutrient, that nutrient is considered deficient. There is the low concentration what's still insufficient, or there's the sufficiency level, and the toxic and excessive level. So I will show this is in a figure, and it will be clear now. So if you have that the y-axis here, it's the yield of the plants, and the x-axis is the concentration of the nutrient, there is a limit where this nutrient is so low that the plants cannot grow really well. And it reaches a point where there's an optimum level, an optimum level of this the concentration of these nutrients, where the plants can perform at its highest yields. At 95% is normally where we define the concentration of nutrient that satisfies 95% of the possibility of this plant yields, the maximum plant yields. This is where we draw the line where we have deficiency and sufficiency. Yeah. So we have this sufficiency levels where we have maximum yields, optimum supply of these nutrients. If we, of course, continue raising this level, we have a redundancy, and we have a luxury supply of this nutrient, and if it's too much, it may cause toxicity to plants. And we're going to talk about how is it that these nutrients may cause toxicity to plants in some cases. Others are very rare. Another relationship that we must talk about is that we're talking about before about the concentration in soils, but this is not directly reflected as a concentrations in plant tissue. It might happen that in extreme deficiency situations, actually the concentration in plant tissues may go up, and you have high concentrations of the nutrient in plant tissue, even though that nutrient is deficient. It means that the plant is just sacrificing growth. The plant is not producing biomass, and then it's concentrating that nutrient a lot. So it means, for example, let's say phosphorus, you find this behavior, and I observed this behavior experimentally before. When you have, for example, that you have very drastic deficiencies of phosphorus, the plant will completely stop growth. A wheat plant will stay this big, for example, and it will try its best to produce seeds. And because it's sacrificing growth and biomass, all the phosphorus that it has is concentrated in the little biomass it has. So you might find that a low range of availabilities where the plants are suffering from extreme deficiencies, you may find that still you have high concentrations of this nutrient. And in the case of high toxicity, the sacrifice of growth because of the toxicity may cause also some nutrients to accumulate in much higher amount. Okay, so the plant performance on the dashed line here, it's not necessarily directly correlated with the concentration of the nutrient in the plant tissue, because in extreme cases, the plant is sacrificing growth and trying to just complete the life cycle as quick as possible. All right, so these are symptoms of deficiency of different elements in different plants. I'm going to show quickly how it looks like. A phosphate deficient plant this is very exaggerated here, of course. This is not like that on the field, but it will show some purple color when you have phosphate deficiency in monocotyledons. Potassium deficiency will show as this chlorosis effect, intervenient chlorosis. and nitrogen deficiency will show as a yellowing, overall yellowing. It's worth saying before we continue that the symptoms of deficiency on plants, they are not a good indicator of the deficiency, the actual deficiency of these nutrients. The reason is some of these symptoms may be confounded with other things, like plant disease, for example. So might think that some of these symptoms they're very clear, but there are some plant diseases that cause exactly or very similar symptoms. So it's very hard to differentiate just by looking. In the case of plant nutrients, the soil analysis is the way that we primarily should focus in evaluating the fertility of these nutrients and how sufficient they are in the environment. So we do soil analysis, and that is a better indicator if these nutrients are sufficiently available to plants or not. So here you see these the symptoms of deficiency for tomato leaves. For example, this is for lettuce also. How they show. Overall, there's less plant grows and there's a different coloring, and sometimes you will see the burning, the burning of the leaves. But for example, here the symptoms, they would look similarly with high salinity, for example. If you have high salinity, you may find similar symptoms with this one. Bean leaves, again, similar patterns that you will find. And here, the next slide, it will show about toxicity of micronutrients. Micronutrients, you may have some deficiency of micronutrients, but most likely what you have is, if you have an excess of them, they will cause toxicity. So here is showing very clearly how boron, molybdenum, manganese, and zinc can cause extreme toxicities to plants if they are over-fertilized or if some soil contain these nutrients in too high amount. Well, the basic nutrients, as I was speaking before, they are not coming from soil minerals. They are coming from water and air. And in general, we can manage these nutrients through irrigation, to water management, and aeration of the plots. So we don't think about these nutrients as fertilizer-related nutrients, but we think about these nutrients more as how do we manage the aeration of the system and the water supply for that system. Too much water supply may also be damaging about the soil air, and too little water supply will be limiting for the water availability to plants. So we're going to have another lecture about soil water and also about the chemistry of nutrients in soil, the general concepts about the chemistry of nutrients in soil, and this will be clearer how these factors are interrelated. So why is nitrogen, talking about the minerals in soil, the nutrients that the plants are taking up from the solid phase, from the soil solid phase. We're going to have a full lecture in nitrogen and all the macronutrients, but in general, why is nitrogen so important? Nitrogen is very important because it's part of amino acid and proteins. It's part of amino acid and proteins. But also because it's integral constituents of the bases that are present in DNA and RNA here in the right. So because it's DNA and RNA is coding for all the proteins in the cells, and the proteins are carrying out the enzymes, for example, the proteins are carrying out every biochemical function on the cell. So altogether, nitrogen is involved in the full plant physiology. There's nothing in plant physiology that nitrogen is not involved with. Same goes for phosphorus. Phosphorus is involved in all plant physiology. Also, it's on the backbone of DNA and RNA, linking the sugars. But also it's involved in ATP and ADP as energy transfer molecules. It's structurally involved also in phospholipids. It's the phospho in phospholipids. In the is what gives the phospholipids its hydrophilic properties. Yeah, you have these phosphate groups on the phospholipids. There are molecular and silane molecules that are phosphorylated and dephosphorylated, and that makes the information of what is the extracellular environment of the plant be transmitted to what genes the cell should be expressing. So there's a lot of molecular signaling that are involving phosphate molecules. And also there's many proteins, especially in eukaryotes, many proteins that are phosphorylated also. So the role of phosphate is a lot more complex than the role of nitrogen. Yeah? And we will talk about this again when we have our lecture about phosphorus. But just as an introductory subject, just mentioning what are the things that these nutrients are doing in the plant, yeah, inside the plant physiology. So potassium, why is potassium important? Potassium, differently from other nutrients, potassium is not found structurally in molecules in plant tissue. Potassium is always found as ions, but as ions, they are really important on the overall balance of these ions and establishing these electrochemical gradients within in between environments that are separated by cell membranes. So this homeostasis, this equilibrium between all these processes that are dependent on electrochemical gradient in the cells, they're strongly regulated by the potassium. So we need potassium in quite high amount in cells. And potassium is very involved in transport of other nutrients and water use through the regulation of stomata activity. And in general, all the processes in the cells that are involving ions flowing through membranes, potassium is highly involved in those. Calcium is basically involved on cell wall structure in the development of new tissues and meristems, root development, enzyme activation. There are several mechanisms and physiological mechanisms that calcium is involved with. Also, together with phosphorus, molecular signaling is important for potassium, membrane stability, disease resistant. It's a quite immobile nutrient in plant tissues. And it's important also as an ionic form to counteract the organic acids. Yeah, normally this is the go-to ion that will complement as a counter ion for organic acids such as citric acid, malic acid, oxalic acids. You need calcium to be the counter ion for these organic acids in intracellular environments. Magnesium have two very specific roles. It's structurally part of the chlorophyll molecule. It gives this property of the chlorophyll that it will vibrate when exposed to some wavelengths of light, and gives the function of this chlorophyll because of this head containing the magnesium. So we will have also a strong role of magnesium on enzyme activation. Sulfur is highly important because it's structurally part of proteins. Basically, it's part of two amino acids, cysteine and methionine. It's highly involved in the conversion of nitrate into amino acids. and then into proteins. It's highly involved in the stability of proteins because of the disulfide bonds that we find in these proteins. It's part of vitamins and really relevant for chloroplast growth and function, and therefore photosynthesis in general. Nitrogen fixation by legumes is highly dependent in sulfur nutrition, and sulfur is fairly immobile in plant tissues. So how do these plants take up these nutrients? Some of these nutrients are cations, some are anions. They have different chemical behavior in soil solution. And not all these nutrients are coming up through diffusive and a convective flow of water inside the plant tissue. Some nutrients need to be taken actively. You need to spend energy to move these nutrients from the extracellular environment into the intracellular environment. So we have two types of uptakes. We have passive and active uptake. On the passive uptake, the nutrients will come inside the cell and they will be moving with the water. Yeah, it will come in favor of the electrochemical gradient. Yeah. So if the concentration is high on one side and it's low on the other side, it will move along that concentration gradient and move inside the plant tissue. So this is passive uptake. In general, as a general rule of thumb, the cations, they will be behaving more as a passive uptake, and the anions will be being used by the plants and uptaken through active mechanisms. So we must differentiate also that the passive uptake can be mediated or unmediated. So there are some proteins that, although they are working in favor of the concentration gradient, they are selective. So the sporins transport proteins on the cell membranes. They are able to regulate that some types of ions can go through, but other ions may not. Yeah? In the active uptake, not even if you have convective processes happening, this ion will not go through because they are working against this concentration gradient. They're working against the electrochemical gradient. So you need to have some transporters, some molecules, some proteins that will use ATP, will use energy to move these molecules from the extracellular environment into the intracellular environment. This is the case for phosphate, for sulfate, and basically most of the anions on the soil system. You will need transporters that will regulate the uptake of these nutrients. So one of the important things that we must say is that these plants may regulate the amount of these transporter molecules up and down if they need more or less of these nutrients. And also the plants may also, some of these channels are shared between different ions. When these channels are shared, it means that if you have too much of one ion, you will decrease the absorption of the other ions. And this will be competitive absorption. Yeah? So if you have too much potassium, you will decrease the absorption of ammonium, for example. So this, even if you have sufficiency levels of some nutrients, you might have a deficiency symptom because of the over-excessive amount of another competing element. That is, they're competing for the absorption channels on the cell membranes. So where does this occur? This occurs on the roots. Yeah. So you will have that the roots. These are different structures for monocotyledon roots and for dicotyledon roots. But actually, these nutrients will be present on the soil solution outside the root, and they will have to diffuse through the cortex. Yeah. And while they are diffusing through the cortex, they may be uptaken to the intracellular environment. And when they are in the intracellular environment, they are passive to be taken by convective flow through the plant vessels. You know? So these absorptions is not only through the cortex. It's important to say that the plants have root hairs. These root hairs expand the volume exploration of the soil. It increases the surface area of the roots, and increase the more plant hairs will be present, the higher the nutrient and water uptake by the plants. And once these nutrients are shifted into the intracellular environment, they have access to move from cell to cell to these structures called plasmodesmata. Yeah, they're linking every cell on these root systems. So the nutrient that is, once it's uptaken to the intracellular environment, and then can move from cell to cell using these plasmodesmata channels. So you have that there are different pathways of the uptake. There is the apoplastic pathway, which is the nutrient is moving through the extracellular environment, through the cell walls, the pores of the cell walls. The second mechanism is transmembrane, where the nutrients are going in and out of the cells on the cortex. And the third is the symplastic pathway, where these nutrients are coming into the intracellular environment, and then through the plasmodesmata, they're moving through, within the cells up to the xylem. But just to finalize this lecture today, we just want to give you a hint that these nutrients on the soil, this is what happening when they come in the roots, but in the soil there are many factors that affect how available these nutrients are. These nutrients must be soluble, and they must be on the ionic form in the soil solution in order to be uptaken by the plants. And there are many factors in the soil that regulates how soluble these nutrients are, and how these nutrients can be available in ionic forms, not in molecular forms. So some of these factors that control the bioavailability of different nutrients in soil are related to the cation exchange capacity. We will talk about that in the next lecture. Clay content, organic matter content, or oxide content. The total concentration of the nutrient is important, but its bioavailability is regulated by other factors. Soil pH will regulate the solubility of these precipitated ions. Some ions will be in precipitated molecular form at some range of pHs, but in ionic form in other range of pH. Salinity may regulate this uptake by plants by competitive absorption of different ions. Redox potential of the soil. There are some also plant factors, biological factors like the upregulation, non-regulation of transporters, or more presence of root hairs, better exploration of the soil volume by roots. There are many factors on the plant side. And we have also microbial factors outside the roots and inside the roots. It's full of microbes, and these microbes, they are negotiating this process of the plant nutrient uptake. So we're going to talk about soil biology also, and how soil biology is affecting plant nutrient uptake. But overall, this is what I have for you today. I hope you enjoyed. In next lecture, we will talk a little bit more about soil chemistry and how it affects nutrient uptake by plants. Thank you very much. See you next video.